Hello, I'm Meredith Blackwell. I'm here at IMC 12 in Maastricht, Netherlands, and it's the 14th of August, 2024. And we're going to have an interview with three people, and we're going to have them introduce themselves.
Okay.
So hold up your cards and
I'll start. Okay. Hi, I'm Maria-José Romero-Jiménez. I'm a PhD candidate at Oregon State University.
Okay.
And I'm from Costa Rica.
Oh, okay.
Well, hi, my name is Diana Vargas Hernandez. I am also from Costa Rica and I am a PhD student in North Dakota State University.
Where?
North Dakota State University.
Okay.
And my name is Millicent Sampson-Eves. I'm currently a post-baccalaureate researcher at the University of Puerto Rico, and I'm originally from California.
Okay, yeah. So we want to know, we know where you were born, what countries probably, but give us a little more detail about exactly where. And then you might even want to go ahead and talk about how you got interested in fungi. And then we'll go from that after all three of you have talked about that.
Sounds good.
Cool. Really, do you want to start?
Sure, yeah, I can start.
Yeah,
So I was born in California in the Bay Area. I grew up there. I went to Berkeley High School. And so I spent a lot of my life around lots of community gardens. I was very lucky to be exposed to that, very young. So I remember I first learned about arbuscular mycorrhizal fungi when I was doing my undergrad at UC San Diego. I was also involved in the community garden there. And so I was interested in their role in crop production in aquaponic systems, which is a little different from like traditional farming.
Yeah.
And then I was studying abroad in Costa Rica, where I was interested in the role of arbuscular mycorrhizal fungi in mangrove restoration areas. And that's kind of how I got interested, and then I just kept going down that path.
So you got interested really pretty much earlier than I did, and so
Yeah.
that's fairly early. So I was just going to say I've just visited the Bay Area. My granddaughter just graduated from Berkeley.
Oh my God,
In
congratulations.
math. Oh, she
Wow.
got a PhD in math. So
Yeah.
it was a pretty nice place. I love the campus.
Yeah, it's beautiful.
Did you know any of the mycologists there, even though you went to school in San Diego?
I can't say I was lucky
Okay.
enough to have met any of
Because
them.
John Taylor and Tom Bruns were very well known, and there are other mycologists there as well.
Yeah.
So,
Okay, well, I was born in Costa Rica. I am from a really small town near to the center of the country. My family are coffee farmers, so I always be exposed to a lot of nature and a lot of agriculture, and I always saw how difficult and how challenges are fungi, as plant pathogens. But also I also always have this a dream or idea or illusion to find ways to try to help the farmers in a way that we can use natural products. I know how difficult are agrochemicals and how expensive they are, how bad they are for the environment. So I'll go pretty early into that. So
did you have this in school or just?
no, it was
just-
I was in a really small town, a really small school. But then I was lucky enough that there is this a high school that is focused in science, and
Oh, okay.
there I got a lot of support and motivation to pursue a STEM career. Actually, that is where I met Maria, and then as undergrad I was pursuing a biotechnology career. So again, it was a lot of using natural products for apply solutions to the real world. I started working in a biocontrol lab with fungi, so that is where I started my journey to get more into fungi. At the beginning, I didn't know how what to use or how I want to achieve to try to help to find more green solutions in agriculture. But then I found fungi and understand a little bit more, and then I got really good mentors, Dr. Andrea Porras Alfaro, that also I was lab mate with Maria, and then now I'm working with Dr. Laura, with coffee and the fight. So it's been this connection in my background in coffee and then fungi, so it's been really
Full circle.
full circle and
Yeah,
really
yeah.
engagement and really a personal a lot of personal motivation in
Yeah.
that area.
And how did you get interested in it?
I took we did the same undergrad, and we took an agriculture class. And during the field work, we had
And,
to
and where was it?
Costa Rica.
Yeah, okay.
Yeah,
You-
biotech engineering.
Okay.
And we took I didn't like the career that much initially because it was like very plant heavy. But we had to take an agriculture class, and we had to grow a plant. So in my case, it was beans, and we would see the plant pathogens. And the professor that was teaching the class like would tell us the pathogens, but then would tell us like, people use trichoderma for these. And then eventually I started working in his lab with trichoderma as a biocontrol agent. And then while I was there, I needed to do my honors thesis, and he suggested Andrea's lab to do my honors thesis there. So I moved to Illinois for 6 months, and then I stayed for my master's there.
Oh, okay.
Yeah.
And how did you get to Oregon State?
Kind of because of Andrea. When I was looking into PhD programs, I was like, okay, I want to do a PhD. I want to work with endophytes. Do you know people? And then she sent me a list of PIs, and I just went through it. And Posey Busby was on that list, so I kind of like Googled her. And then at the MSA in Minnesota, I talked to a couple of her students, and I was like, oh, okay,
Yeah.
I like what they're doing. I like their environment. And eventually I talked to her and made the transition over there.
Okay, and I have a connection with Oregon State, you
Joey?
know?
Yes, yeah, Joey's on my committee.
Tell me something about Joey. He's
Yeah.
my former PhD student, my first one that really went on. Does he behave? Is he a good
Yes.
mentor?
Yes.
Is he
Yeah,
good,
yeah.
Is
No,
he a good
yeah,
department
yeah.
head?
Yeah, he's a great department head. I think that he really listens to the grad students, and I really appreciate that because I don't think that that's the case for all departments. So he does have a great communication
Yeah,
with the grad students.
he's good. I think what was it, Moldy Mondays? Did he start a seminar? They
I
may
think
not
so.
still have it.
I don't think we have it, but there's still stuff happening. So.
But
It's
you're
still
lucky
fun.
because you're in a department where there are a number of mycologists, which is good. Yeah. So, what are you working on exactly for your?
I'm working with coffee endophytes as
Okay, so
potential.
you're isolating them, or
Well, some collaborators are isolated for me, and now I'm
Okay.
testing their potential as biocontrol agents against Mycena citricolor, that is a coffee pathogen. Yeah.
So when you go home, do you isolate some yourself from the field?
Yeah,
or?
we have one of our field sites is in Costa Rica, and I have isolated a couple from
Okay.
the town that I am from. Yeah, so it's been nice, and it's really an integrated experience because like in the lab you are so used to use like the isolates in plates, but right now there is a really important outreach of Mycena in the field. My dad is a farmer, so he sent me the pictures of like, oh, look how it looks
Okay.
and how it's going, or oh, look how the coffee plants are looking and stuff. So it's been really, for me, it's connecting that experience of like my professional career, like as a mycologist, and also have the like what is happening in the field every day. So
Yeah.
it's been
So Mycena,
Mycena.
and it grows. How does it grow? Is
It grows in the leaf, so
okay?
you can
So
see.
it's a parasitic one.
Yeah, it's
Yeah.
pathogenic,
Oh,
it's
okay.
parasitic, and it reduces a lot of the yields of the coffee plants. So we lost a lot of like, especially in rainy season. This year it's been raining a lot, and it's very humid. It's been a lot of like outreach of the Mycena. So you can see like the little like fruity body in the leaf. And yeah, it's
Does it form rhizomorphs? Rhizomorphs? Does it form rhizomorphs? The Mycena?
I don't think
No,
so. I
okay.
think that it's like
That's marasmius, I think, that does those.
Yeah.
it forms like a yellow.
yeah, it's
spot.
like a
Sclerotium,
yeah,
or-
exactly.
It's
Or
like a yellow
mushroom?
spot, like very
Oh,
small.
okay. Oh,
They,
Yeah.
okay.
they call it rooster's eye.
Yeah, exactly, because it looks
Looks
like,
like-
like the rooster. So you can see the lesions in the leaves, and then sometimes even the fruity body comes from the
Yeah.
lesions. So it's really interesting to see that connection, and especially like here, you see like a lot of people talking about mycena, but it's not that well known that it can be a pathogen.
Right, that's why I knew about marasmias, some of the marasmias, because in Louisiana we have rhizomorphs that hang down from the trees. And it was a long time before anyone, well, people knew, but I remember having one sent to me in my lab, and it had been all around the country. And somebody said, oh, it's the inside of Spanish moss, which is sort of a black strand. But anyway, I knew because I had looked. And I flamed them, the marasmias rhizomorphs. I flamed them and then put them on agar and got a mushroom out of it. But, so I just wonder if you could do something like that with the leaves, if you could put them in culture somehow and induce the mushrooms to form.
I noticed, like, even in PDA, sometimes you can see some, like, a little, like, heads, little spots that start forming, but not the whole fruity body. Yeah.
So how do you know what it looks like? Do they have it in Puerto Rico too?
Well, I was actually part of the same lab as Diana. In the gap between my post-bac and when I graduated from undergrad, I was working with Dr. Aldrich Wolf at NDSU, but we went to Costa Rica to do field work.
So what an experience.
Yeah, so I was there for about a month helping with collection of leaf samples and roots and counting harvest. Yeah.
So right now in Baton Rouge, where I was, there were some people that were in Costa Rica for many years that worked with the Smithsonian, and then the husband did Smithsonian and the university. But they were interested in some of these diseases, but they came to them late. But I know that they did a lot of work with students out in the field and looking for fungi. In one case, insect-associated fungi.
Yeah.
But it's so. What are you going to do now?
So right now, I'm doing the post-bac in Puerto Rico for a year. And I'm working with Dr. Fristow, who is not associated with mycology. He does invasion biology and macroecology. So I'm currently just learning more about data science because I'm really fascinated in like how we can like utilize powerful data and the information that we already have, trying to compile like complete databases so that this information is accessible to all of the researchers around the world that need it in a way that, you know, makes sense, because it's sometimes, especially with all the taxonomy name changes and with fungi, it's like, especially for like more cryptic species, it's hard to know like whether they're native or alien in certain geographic regions, and like how to track invasions. So that's currently what I'm trying to learn more about, but through the mechanism of plants. You
can certainly apply that to fungi later if that's what you choose, because you know, I've always been impressed with the number of species of fungi that were unknown until they invaded in a new place and got names.
Yeah, exactly.
And I guess we talk about the Dutch elm disease, which is Dutch only because they described it here. Figured out what it was. So you've actually got a lot of research done?
Yeah, for my PhD I'm working with yeast of poplar leaves. So describing the diversity of yeast that we can culture from the leaves, and then using them as model systems to understand diversity, disease protection relationships.
Okay, are they Basidiomyces yeast or Asco?
Mostly Basidiomyces, a couple of Ascomyces, but mostly Basidios.
Do they pop off their spores so that they're easy to isolate?
They're easy to isolate, and then you find Arobasidium, like always, and a couple of Phylobasidiums. But yeah, they're.
We used to do a lab experiment, and you would put it in the top of, put the leaf, glue it to the top of a petri dish, and then they're blistosporic. So I don't know if you're.
I haven't tried that, but I'll definitely do
that. You ought
to
try it, because if they're blistosporic, they'll just shoot down, and it's very clean to isolate them that way.
Okay.
If you're careful not to crumble them as you're putting them up there.
Okay, that's good to know. Yeah, I'll definitely try that, because we've isolated them, but just the normal streaks.
Why clover?
What?
Why clover?
Oh, poplar. So Populus trichocarpa. So it's like black cottonwood. Yeah.
Oh, okay. I see.
Yeah. Poplar. It's I think that is the first tree that got its genome sequenced. So there's like huge resources, genetic resources.
Yeah, so that's, yeah. Yeah. So we know what you're doing. You're working on a PhD now.
Yeah.
What are you going to do?
I just started, I
You
was
just-
doing my master.
Right.
I was working as master student in Dr. Albrecht Wolf lab, and recently I'm switching to a PhD. So I just started in the.
So you're going to skip your master's.
Exactly. I'm been working with her the last 2 years, and I'm gonna use that as a base for a master. So I'm kind of like in the middle
Of the process.
Of the process. Yeah.
So you're committed to staying in that awful climate. Is it very different for you? Surely.
Well, yeah, yeah, it's different. It's different. Definitely, it's different. It's a challenge. Well, Costa Rica is a tropical country, and
I
then
know.
coming to North Dakota State University is in Fargo. This is pretty north in the states, and it's really windy and cold. Winter can be like around 6 months.
Oh, I know.
So it's been definitely like a shocking learning
Well, later
process.
on, you can consider it a good experience.
A good experience. That is why, like, at the beginning, I wasn't like, when I first started working with Dr. Laura, she offered me to do the PhD right away. And I wasn't sure for the same reason,
Yeah.
right? Because there is a lot of like, it's not just like the professional develop, it's also like how you feel outside the lab when you are like, you know, like the cultural experience, the living, the whole living situation. So at the beginning, I wasn't sure about it, but at the end, I decided that I'm doing something that I really love and I really engage. It's something that is really, I feel really attached to my project in a personal and professional way. So I decided that, and I have made really good friends in Fargo, so I feel like, okay, even the weather sometimes doesn't help. I still like, coming from a tropical country, the snow is like magical for me, and I get like the experience.
Well,
there.
and also, you know, it's always good, I think, to work someplace where you don't want to live because they don't want to hire you where you're working. So.
Yeah, exactly.
So Puerto Rico, how's that compare with the Bay Area?
You know, very different. But I think I like warm climates. You know, I spent 4 months in Costa Rica, and then I was in North Dakota, and then back to Puerto Rico. And I think the cold in North Dakota, even though it was a very mild winter, apparently,
It was.
I still thought it was very cold. Is nice. And again, like, I really enjoy like the people I've met in Puerto Rico and the culture there, and also what I'm researching. And so, like Diana said, that makes
Which campus are you on?
Rio Piedras.
Okay.
Yeah.
Yeah, one of my colleagues who don't look now but is standing over to your left was in Maria-José.
Oh, Maria-José,
For a number of years before he went to Harvard.
Nice.
But anyway, so when are you going to have to apply for places, or are you're planning to go on, I assume?
Yes. So I'm currently starting, or in the process of applying for grad schools for this current application cycle for the fall of 2025.
And how are you choosing? Are you looking at places or people, or?
I'm considering all of it. So I have kind of decided that I want to, since, you know, I'm thinking about probably doing a PhD, so I'm trying to pick a place near my family. That's something that's important to me. Like, I've done a couple years of like traveling, moving around, and so I think for the long term, that's an important
Well, you have
factor
a number
for me.
of good schools to pick from.
Exactly,
Won't be a
yeah.
problem.
So, and
Yeah.
I've met some people here. My family currently lives in Washington, and Washington is great for fungi. It's the perfect climate. There's a lot of great research happening at the University of Washington, so I'm excited about the possibility for opportunities there.
That's great. And yeah. And how did y'all get together at this meeting? Or did you know you all knew each other? You two knew each other from North Dakota. You two knew each other.
Yeah, and
From
I
Costa Rica.
connected.
So
and-
you're in the middle.
Yeah, exactly. So I just talk and organize it. So we decide like just to come and
Oh,
stay
neat.
together. So yeah, that is
Yeah.
how we.
Were you able to get travel money in the positions you're in?
Yeah.
Oh,
Yeah.
that's great.
Yeah.
Yeah,
Great,
it's incredible.
great. Yeah.
That's a
Yeah.
really great opportunity. So
Yeah.
And so when you finish, what do you want to do? Do you want to?
I'll probably do a postdoc, and I would like to stay in academia.
Okay, and go home or here or?
It would be great if I could go home, but research is kind of limited in some degrees, like money-wise, and then getting supplies. Like if we wanted to order primers, it would take 3 months. So
Yeah.
in Oregon it takes 2 days. So it could be in the States. We'll see what happens. Time will tell.
Okay. And I was going to suggest too. Several people have told me that they pick major professors by looking at these interviews.
Yeah.
So
I'll have to look back at them.
I would pick someone first and then see if we have the interview and then check them out that way until you can get to visit. So,
Yeah.
So do you have any more comments to make about anything or ask questions or?
I don't know.
Have you been having fun?
Oh
At
yeah,
the conference, yeah.
I'm having fun.
Nice.
This is awesome.
But I'm not hearing any talks, but I will get to go to talks tomorrow morning.
Awesome.
You're
Nice.
gonna okay. That's nice.
That's exciting.
How many people had you interviewed these days? Do you know?
33 or something?
33. Wow.
Yeah. So we have 79 interviews from the last IMC, and then 2 MSA meetings, Gainesville and Flagstaff.
Flagstaff.
No, wait, Gainesville and I'm trying to remember
I
now.
have no idea.
So
Okay.
there's
But-
Flagstaff, Gainesville,
Gainesville,
and then this year was Toronto.
right, and we didn't do it in Toronto.
Okay.
Yeah, because we did it here instead.
He's waiting.
So,
Well,
okay.
Thank you so
Yeah,
much.
thank you
Okay.
for interviewing us.
And we
Yeah.
have enjoyed talking to you.
Yeah.
Thank you
Thank
so
you so
much.
much.
Thank you.